Thank you to the organisers. My name's Amy Howard, and today I'm going to be talking about our recent work on the BigMac dataset, linking microscopy-derived microstructure with MR signals throughout the macaque brain. Our ability to characterise the connectome has been greatly advanced by the scientific community's access to big data. But there are multiple ways in which data can be big. Firstly, it can look across the population for multi-subject variability, such as in the 1,200 participants scanned in the Human Connectome project. Alternatively, big data can act to characterise a single brain in exquisite detail, a beautiful example of which being the MyConnectome project, where a single subject was scanned 3 times a week for over 50 weeks. In the macaque, recent exciting work by Autio devises an HCP-style protocol to both improve data consistency in population-based macaque studies and provide a link to the human HCP data for cross-species analysis. But to characterise the connectome of a single macaque brain in exquisite detail, we present the BigMac or BigMac dataset. The dataset includes in vivo MRI, postmortem MRI, and multi-contrast microscopy in a single whole macaque brain. The in vivo data was acquired over multiple scan sessions. There is around 270 hours of postmortem MRI and over 1,000 hours of microscopy, resulting in several terabytes of raw data. So let's unpack BigMac a little. The in vivo MRI includes structural MRI, task fMRI, resting state, and diffusion data. The postmortem MRI includes structural MRI, T1 maps, and an extensive diffusion protocol. This provides us with an excellent opportunity to link our postmortem results to in vivo data in the same single brain. The diffusion MRI data includes 2 spatial resolutions, high-resolution 0.6mm data at a b-value of 4,000, as well as 1mm data at b-values of 4, 7 and 10,000. Here, the 0.6mm data is able to characterise single fibre populations with fewer partial volume effects, whilst the 1mm data allows us to push to higher b-values whilst retaining good signal-to-noise ratio. Further, at 1mm we have both linear and spherical tensor-encoded data to facilitate more advanced microstructural modelling. Finally, at 1mm we were able to acquire 1,000 gradient directions in the outer 2 shells in a reasonable amount of scan time. And of course, with high b-value, high angular resolution data, we obtain good diffusion contrast. Here I show diffusion MR images from different gradient directions as we trace a spiral over this Q-space sphere. All postmortem MRI data was acquired on a 7 Tesla small animal scanner with a spin echo multi-slice sequence with single line readout. This sequence minimises image artifacts but is fairly slow, resulting in the 270-hour scanning time. So we want to link the MR signal to microscopy data, which has both ultra-high resolution and high specificity. But what microscopy data will be included in BigMac? Following MRI, the BigMac brain was sectioned along the entire rostocaudal axis with consecutive slices processed for polarised light imaging and histology. Each microscopy section is repeated every 350 microns, and we have full brain coverage. Here are example images of the microscopy data we have acquired so far across the entire brain. Polarised light imaging utilises the birefringence of myelin to estimate the primary fibre orientation per pixel in 2D. The histology data is acquired with sub-micron resolution to visualise both myelinated fibres, as shown in the middle where the myelin is stained brown, and cell bodies from the Nissl-stained on the right where the cells are stained purple. We can then process the histology images using structure tensor analysis, a type of texture analysis, that allows us to estimate the primary fibre orientation per pixel. Here on the right I'm showing the fibre orientations estimated from the myelin-stained slides. However, we're not limited to running structure tensor analysis on only the myelin stains. On the left here I'm showing the orientations estimated from the Nissl-stained slides, where the cell bodies tend to align with the orientation on the fibres, as shown on the bottom. Here we see very similar orientations from both the cell bodies and the myelinated fibres, where both are even able to resolve these intermingling fibre bundles within the corpus callosum. The multimodal BigMac data was then co-registered using TIRL, a new MR microscopy registration tool developed by Istvan Hazar. Using TIRL, we can map each 2D microscopy section into the 3D MR volumes. Here I'm showing an example output where I've co-registered a PLI image to the structural MRI. On the structural image, the green lines indicate the white grey matter boundary, whilst the orange lines show the tissue edge. When I overlay these lines onto the PLI image, we can see close alignment of the data. This facilitates meaningful MRI microscopy comparisons at the voxel level within BigMac. We believe there are many applications for the BigMac dataset, including neuroanatomical investigations, multiscale neuroscience, diffusion MRI methodology and model validation, as well as opportunities for machine learning and data fusion. However, today I would like to highlight one example analysis afforded by the BigMac dataset. This analysis is inspired by the recent acquisition of Nissl-stained histology in BigMac, where we can easily visualize the cortical layers due to the difference in cell body density, as well as orientational information from glial cells in the white matter and to some extent cell bodies in the cortex, which tend to align with the expected fibre orientations. So the aim of this work is to utilise BigMac data to link microscopy-defined microstructure with MR signals in the cortex. Our method utilises the natural organisation of the cortex to extract a series of cortical profiles which extend from the white grey matter surface to the cortical edge. This is a powerful method for multimodal comparisons, as these 1D profiles provide both a rich description of the data and a common axis along which MRI and microscopy data can be meaningfully compared. To extract these cortical profiles in BigMac, we generated surfaces between the peel and white matter using a modified free-surfer pipeline. This resulted in a series of matched vertices, which together describe a set of profiles across the cortex. To compare matched profiles from both MRI and microscopy data in the same brain, we first needed to map our profiles, which were initially defined in MR space, onto the microscopy data. This was done using TURL, where on the left I show preliminary results where the cortical profiles have been mapped onto the microscopy plane. Various MRI and microscopy metrics were then sampled across these profiles, where in this early analysis we analysed over 280,000 profiles per contrast from the left hemisphere. On the right, I'm showing the average profile from the red region of interest. The results I'll show today include postmortem MR metrics, such as the structural MRI intensity for multi-gradient echo sequence, as well as fractional anisotropy and mean diffusivity from diffusion tensor analysis of B4000 data. Our microscopy metrics include the Nissl-stained density, a proxy for cell body density, and microstructure orientation dispersion, which is a measure of how coherently oriented the Nissl-stained microstructure is. This metric was extracted after structure tensor analysis of the Nissl-stained slide. Here are our first results, where for each contrast we take the mean profile within each region of interest of the CHARM3 cortical atlas. We then correlate the mean profiles on a region of interest basis. The histograms summarise the range of correlation coefficients across all regions, and the red line indicates the mean. We find that the structural MRI correlates negatively with Nissl density and positively with Nissl dispersion, and that these results are fairly consistent across the cortical regions. In comparison, the diffusion tensor metrics show generally less consistent patterns, with some regions showing strong positive correlations and others negative correlations. This demonstrates pitfalls in validation studies which compare metrics across limited regions of interest without whole brain analysis, as you may come to different conclusions if you consider two different regions. So in these results we simply compare the two modalities, but can we do more and instead simultaneously combine them into some sort of joint analysis? This next set of results uses canonical correlation analysis to look for common modes of microstructure variation across the brain. The plot shows the correlation coefficient of the first component. We find a common axis of microstructure variation between Nissl density and structural MRI, which is reassuring as it mirrors recent results found in humans. We can then map this component back onto the CHARM atlas, where in these preliminary results we see a clear pattern separating the frontal, occipital and parietal temporal areas. Notably, the DTI metrics show only a weak correlation with the microscopy, suggesting that structural MRI but not DTI metrics provide sensitivity to brain-wide variations in Nissl-stained microstructure. To conclude, today I've introduced the BigMac dataset, a resource combining in vivo MRI, postmortem MRI and multi-contrast microscopy in a single whole macaque brain. Future work will focus on more advanced multimodal comparisons which utilise the wide variety of contrasts in BigMac, including more microstructurally specific metrics from diffusion MRI, myelin-sensitive microscopy, and in vivo signals. With that, I'd like to thank all those involved and thank you for listening.